Okay, everyone. So now I'm going to tell you about some of my team's work and how we are working to collaborate with the MJD community within Australia to develop some drug testing studies. So our work investigating MJD started from an idea of Professor Garth Nicholson. So Garth is a geneticist and a neurologist, and he travels quite a lot. And so apparently it was through his travels within Australia that he had started to come across people living within northeast Arnhem Land of Australia here, who he noticed were developing neurological symptoms of that appeared to be caused by a progressive neurological condition. And because of the prevalence and the number of family members affected, he came to suspect that perhaps it was a genetic condition. So Garth was also involved in the initial genetic diagnosis that, yes, in fact, these patients were developing a genetic disorder and that it was MJD. So following that, in around 2008 to 2009, it was with great foresight that members of this community and through their governing representatives, the Anandiliaka Land Council, including Gayangwa, who is here, made a decision to put some of their own money towards researching for a possible treatment for MJD. And that was then facilitated through work of the MJD Foundation to open up a grant scheme to fund potential research into MJD. And Professor Nicholson actually put forward an application to develop a zebrafish model of MJD to use in drug testing studies. And it was successful. In around that time, actually, around 2009, I first met Garth at a motor neuron disease conference, and I was working as a zebrafish researcher, as a neuroscientist, using zebrafish to study motor neuron disease. And Garth said to me, "You have to come home and help me with this project." And of course I obliged, because it sounded like a very important condition, and I was incredibly homesick living in Belgium at the time. So at this point, I'm sure that many of you in the room may be asking, but why would you use a zebrafish to study a human condition? And there's actually quite a lot of reasons, so I'll take you through some of them now. So these include that zebrafish are actually easy to get large sample sizes for our studies. So zebrafish are very small. They're only about 2.5 centimetres when they're fully grown at adult stage. So we can house thousands of them within one room. And a female zebrafish actually has hundreds of offspring each week, which means that we can also get large study sizes for our treatment studies. Zebrafish also are vertebrates, and they develop most neuroanatomical structures that humans develop as well. They are transparent during development, and you can see here the nervous system of a zebrafish in the living animal. So this means that we can actually track the development of motor neurons or various neurons, and also we can track proteins that are expressed if we give them a fluorescent tag, all in the living animal. They also are easy to genetically manipulate because they develop outside their mother. So this means that we can inject into a embryo to make a transgenic model. So we add DNA to do that. And they develop quite rapidly, actually. They have movement within the first day after the egg is fertilised, which of course means that examining movement related to movement disorders can be done quite rapidly. But really my favourite reason to use zebrafish is that we can perform drug testing studies relatively easily because we can add drugs to the water that they are swimming in. So it seemed like a good idea to go ahead and develop this transgenic zebrafish model to study this disease. But how did we do this? So really developing this project and continued work on this project has occurred through a process of ongoing consultation with the MJD patients living within Australia. And the way that this has worked has been a combination of us researchers talking to the MJD patients and the community that is affected within Australia about our plans and our progress. And this community also talking to us about their ideas or their requests, and sometimes even their concerns. So what does this look like? Well, first of all, in the beginning of the study, it was myself and my research assistant here, Christy, who's still working with me, and we were just surrounded by thousands of zebrafish that we were developing this model. And from time to time, we had different members from the community in Northeast Arnhem Land come to visit us to touch base on what we were doing and to take news back to the community. And this included visits from Tony, shown here, Gayangwa, who's here today, and also Joyce, who's here today as well. And these visits continued over time. And in 2016, we moved to Macquarie University here. And we've still continued to have visits here from Mary and her granddaughter, and Bacala, whose movie you will see later on. But I've also visited Groot Island to discuss the project and to talk to community there about what we were doing. And more recently, as you can tell, I've changed a bit over the years, but more recently also visits to Darwin. And these discussions that we've had have taken place in all sorts of situations, including here, where 1 minute Gangwa is teaching me how to do basket weaving. And then the next minute, Bacala turns up and says, Angela, you have to tell me more about this research that you're doing. I think during this visit, I prepared a presentation and I gave that presentation on 6 different occasions, often one-on-one with different people who had requested me to explain what we were doing. So how have I explained this to the patients? Well, this is one slide. This is just an example of how I have explained some of our work. And it included that, the aim of our work is to look for treatments that might improve the movement of the zebrafish. And what we would be testing would be drugs that either are already approved for treatment of other diseases or new drugs that we don't know much about. I've told the patients, and in these discussions we discussed that I can't guarantee that I will find a treatment for this disease, and I can't say how long the search is going to take either. And certainly it's important to know that anything that we find in the zebrafish will need to be confirmed in other models of the disease before they could be used by patients. So what does our work involve? So first of all, we had to develop the transgenic zebrafish, and that involved taking the MJD gene, either with the long CAG repeat that would lead to a MJD phenotype, hopefully, or the short CAG repeat to serve as a control. And we can inject that into the zebrafish embryos and then watch as they develop in order to raise them to adulthood so that we can mate the animals and see whether this gene has been incorporated into the genome and therefore transgenic animal has been developed. So it takes about 2 months for this process to occur. And then after that point, we were able to see that we had some transgenic zebrafish, and we were able to make what we call a colony, so a whole family of these animals. So now that we have established this model, I'm going to show you also what it looks like for the day-to-day work that we do now using these fish. So here's a video where you can see some of our tanks. We have hundreds and hundreds of tanks. And here's Maxine, who's doing some setups to breed male and female zebrafish, so that then in the morning, male and females mate, and the eggs will drop to the bottom of the tank. Then they can be looked at under the microscope, and we can check whether they are carrying the coloured protein that gives us an idea that they are carrying the MJD. And this image here shows you that using zebrafish, because they are see-through, we can also see structures that we have given colour to. So for here, the nervous system is red, so we can see the spinal cord and the axons of neurons coming to the brain and the eye. And that allows us to also study those anatomical structures. But as I mentioned, it's the drug testing studies that we're most excited about. And here you can see Maxine distributing zebrafish embryos or larvae that have been treated with drugs into a multi-well plate. This is at day 6 after a treatment period, so that they then can be shifted into the tracking machine, which you can see here. This allows us then to record the movement of the animals in response to a light stimulus. And you'll see here their response. It's a bit fuzzy in this image, but you can see red lines turn up if they're travelling fast and green lines if they're travelling slow. But luckily, it spits out a distance for each animal, and so we can just look at whether any of the drugs have increased the distance travelled. We can also use this machine to monitor the movement of the adult zebrafish. So you can see them swimming here as well, using the red is fast movement and the green is slow movement. But we tend to find that we would like to focus on the zebrafish in the earlier stages, because that's the advantage of this model, is that it can be very rapid for testing to get ideas of what things might be useful for testing in other studies. And we feel that the adult studies are probably more suited to other models such as rodent models. So in summary, this is how the model looks. We have the MJD zebrafish with the long polyQ length, and they swim shorter distances to the control animals. And so this means that we can go through a process of mating, checking the next day if they're positive, adding a drug, waiting till day 6, and then checking the movement of the animals. We then also make up protein samples to allow us to examine whether different proteins are expressed. But the thing is that this, then we had to decide what treatments we would test. And so we've embarked on testing FDA-approved drugs that we might repurpose, including some that are thought to be autophagy inducers that might release the protein quality control, might induce the protein quality control pathway. Inhibitors of calpain inhibition, which is what Maxine did her PhD on. And Maxine's actually going to tell you about this autophagy work as well. HDAC inhibitors that I'll tell you about in a little bit. And also novel compounds that we have come across through our studies on other diseases, or that collaborators have suggested to us that we should examine. But it is through our ongoing conversations with the MJD community and the community around them that we have realised that it's not just these drug treatments that are interesting to these patients, to our patients. Patients on Groote Eylandt and patients within the Indigenous community have a strong belief also in traditional medicines. And this is something that I think is important for my team to respect and be open to, because, well, not just because natural products have led to treatment of some other conditions, but also for a few other reasons, and I'll take you through those here. So for those that don't know, within Australia, patients of Aboriginal and Torres Strait Islander descent actually have a shorter lifespan than the rest of the Australian community, and it's actually 10 years shorter. And there's a range of reasons for this, and this includes predisposition to different ailments, but also decreased access to healthcare services, and also due to a lack of confidence in perhaps some healthcare services. And so within Australia, there is a large amount of work being done to what we call close the gap, so decrease this difference. And some research has indicated that actually having practitioners listen and be open to the traditional medicines that this community is already practicing may be beneficial in also having these patients or this community embrace other types of medicine. And that might also be useful for perhaps future clinical trials that may develop from people's developments, hopefully a future treatment. Also, patients within this community are apparently also using traditional medicines already, so it's important for us to be mindful of this. And also, as Jen and Joyce and Gangwa's talk already showed earlier today, the patients feel that searching for good medicine is helping them both on the inside for their well-being and their connection with their community, and also on the outside through increasing their physical activity. So for that reason, we're also now embarking on starting to test some natural product extracts that are requested by the community, and also testing compounds within a library of natural products. But this means a very large number of drugs to test. And for that reason, we now have expanded to incorporate also cell culture model testing, which we're trying to make as high throughput as possible. So see Katie's poster regarding this in order to confirm hits in the zebrafish, and then take into mouse model through collaboration with Patricia's team, which is a great progress for us. So I'll stop now, but don't hesitate to get in touch if you think that we could collaborate on this pipeline, or if there's anything you'd like us to test. And importantly, I need to thank the Anandiliaka Land Council, the MJD Foundation, the researchers, and also the community members that have helped with this project. Thanks.